How many different amino acids are there, ma?
20, sir.
20. Okay. What make a difference between one amino acid and the another amino acid?
The R group.
Okay. How many different types of amino acids are there?
20.
No. We classify into how many groups? All 20.
Acidic, basic, and neutral.
Or aromatic, or otherwise also called as what?
Aromatic.
What is this bitter ion?
Uh, it's the neutral amino acid, sir.
Here
It has
acidic
both a
charge.
positive and negative charge.
Okay, very good. Fine. Done. Okay, sir. Another student.
Yes, ma'am. Ranika?
Yes, sir.
Okay, ma. So can you name one example of 16 carbon fatty acid saturated?
Sir, is it lecithin, sir?
Lecithin is a phospholipid. I am asking about fatty acid.
Palmitic acid.
Yeah, it is palmitic acid. So which is the phospholipid that is present in the cell membrane?
That
Just
is
now.
lecithin.
Yeah, it is lecithin. Okay. Which has the high melting point? Saturated or unsaturated?
Saturated ones.
Saturated ones are having high melting points because they're from what source? Animal source.
Amino acids.
Okay. Yes. Good. And can you name another example of fatty acid with 20 carbons?
Arachidonic acid.
Okay. Arachidonic acid. Fine. Good. Done, sir.
You want to ask, you're asking more questions, sir?
Sufficient, I think. So in the next
Okay,
topic,
sir.
I'll ask another
Sure,
two.
sir. Sure, sir. Sure. Okay, sir.
Yeah, they are on the track. No problem.
Sure, sir. Okay.
Okay, fine. So the next is the nucleic acid. So before we shall commence with the nucleic acid, I also request you to go through the structure of cholesterol, which is there in the NCERT textbook, because they may ask you the diagram based questions. Okay, and that will be useful for you if you see the structure of cholesterol. Made up of 4 rings and one side chain. Made up of 4 rings and one side chain. Fine. Nucleic acids are basically DNA and what? RNA. DNA is the deoxyribonucleic acid, and RNA is the ribonucleic acid. What is DNA? Deoxyribonucleic acid, and RNA is ribonucleic acid. Both DNA and RNA are made up of 3 components, students. One is the nitrogenous bases, right? Sugar and the phosphate. So nitrogenous bases are also of 2 types: purines and we call pyrimidines. So we call purine and what? Pyrimidine. Purine is made up of a single ring. Okay. Sorry, purine is made up of 2 rings. Purine is made up of how many rings? 2 rings. Altogether, it contains 9 carbons. Pyrimidine is made up of one single ring, made up of how many carbons? 6 carbons. So, which are the purines that is present in DNA? So, in DNA, the purines are adenine, represented as A, guanine, represented as G, and pyrimidine in DNA is represented as cytosine, represented by C, and thymine represented by T. So everything is same between a DNA and a RNA, except there is one small change in the pyrimidine of RNA. In RNA, one small change you should note. In RNA, the thymine of DNA is replaced. Okay, the thymine of DNA is replaced with what? Uracil. The thymine of DNA is replaced with uracil. Make a note of. Just a minute.
Yes, students, if there is anything that you need to clarify, please talk to sir. If everything is
Please ask.
all right, he can proceed.
No doubts then. Okay. If you did not respond, then it is understood. You don't have any doubts. Okay, we call metabolites. Metabolites are broadly classified into two types: primary metabolites and secondary metabolites. Nothing to explain much here. Primary metabolites is produced by a plant for its own survival. What does primary metabolites include? Carbohydrate, proteins, lipids, and nucleic acid. All these are the primary metabolites. We call primary metabolites as what? No doubt biomolecules. Then what are the secondary metabolites? Why it is produced by plant? Secondary metabolites are produced in plants for its defense mechanism. Secondary metabolites are produced in the plants for its what mechanism? Defense mechanism, which includes alkaloids, flavonoid, rubber, essential oil, resin, gum, spices, and many more. So, since plants are non-locomotory and it is fixed, it has to prevent itself from the herbivores, from grazing, including the insect and the pest. So, they produce the secondary metabolites for its own defense, but we have identified the commercial importance of the secondary metabolites and we are harvesting for our economical benefit. Okay. And this is what you should know regarding the primary and the secondary metabolites. So, what are metabolites? They are the organic compounds including amino acids, sugars, etc. Basically produced through metabolic activity. It's of two types, primary and secondary. Next. See, primary required for survival, basic metabolic processes. What is derived from primary is secondary, produced in more quantities. Secondary is always produced in less quantities. Primary found in all plant, secondary found in only some plant. They are the basic molecular structures. They are not a part of molecular structure because they are required for defense mechanism. They have a physiological role for respiration. Through respiration, what do they produce? ATP. They do not have any physiological role because they are only required for what? The defense. Okay, since they are only required for what? Defense. Examples of primary metabolites are amino acids, carbohydrates, nucleotides, fats. Secondary, alkaloid, rubber, pigments, essential oil, antibiotics, others. Next. This table is damn important. Many times I've asked in the examination. Example of secondary metabolites: pigments, carotenoids, anthocyanins, alkaloids, morphine, codeine, terpenoids, monoterpene, diterpenes, essential oil, lemongrass oil, toxin, abrin, resin, lectin, concavalin A, drug, vinblastin, curcumin, polymeric substances, rubber, gum, and cellulose. Next. So all this we have already discussed, students. So I'll just read it and go through. So what are biomacromolecules? Molecules that have the molecular weight ranging above 10,000 Dalton are found in acid insoluble fraction or pellet or retentate. Everything means the same. Biomicromolecules, they have molecular weight less than 10,000 Dalton, they have mentioned. Okay. So that is generally 18 Daltons to 800 Daltons only, or we can say less than 1000 Dalton. Okay. Next. They are found in acid soluble fraction or what? Supernatant. All this we studied already. Next. Micro and macro difference: small, large, low molecular weight, high molecular weight, simple structure, complex structure, soluble, colloidal. Examples of micro: water, mineral salts, gases, amino acids, sugar, lipids, and nucleotides. Only monomers. But in macro, organic polysaccharides, they include polymers with more molecular. Next. So this is the explanation what I gave you. Why lipids, even though they have the molecular weight less than 800 Dalton, it is included under macromolecules or what fraction? The pellet fraction. Lipids form structures like cell membrane. When we grind, these membranes, they break forming vesicles. These are the vesicles that are not soluble in water. They are separated in acid insoluble fraction, that is pellet, with macromolecular fraction, that is with macromolecules. The same thing I explained to you earlier. Hope it is clear for you. Next. This is a relative composition of cells and its percentage. Water 70 to 90% in a given cell. Protein 10 to 15%. Carbohydrate 3%. Lipids 2%. Nucleic acids 5 to 7%. Ions 1%. You should know this again, this for NEET. Next. 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 Okay. Empty slide. Empty slide. Okay. Listen, students. We have proteins and its biological organization. Let us see. Just now I told you protein is a polymer. Made up of amino acids, which are monomers. How one amino acid join with another amino acid? One amino acid join with another amino acid with the help of a bond called as peptide bond. So which is the bond that join one amino acid with another amino acid? Peptide bond. How? Let me take an amino acid NH2 R H COOH. Let me take another amino acid NH2 COOH H and what? R. Sorry, H and R. Now the reaction takes place between the carboxylic acid of one amino acid with the amine group of another amino acid, resulting in C double bond O NH, which is called as peptide bond. This is what you should know. So which is a bond that is responsible to link two amino acids together? The bond that is responsible to link the two amino acids together is called as what bond? Peptide bond. So when you draw a structure of protein, I draw a linear chain representing NH2 and what? COOH. What does the line represent? Amino acids joined with each other. Okay, amino acids joined with each other with the help of what bond? Peptide bond. See, in the first amino acid, which functional group is free? Amine. And in the last amino acid, which functional group is free? Carboxylic acid. That is what I have represented here. The first amino acid, amine group is free, and the last amino acid, carboxylic acid, is free. And in between these two, the length of amino acids are joined with each other by what bond? Peptide bond. And this is also the primary structure of protein. This is also what of protein? The primary structure of protein. Is it clear? Next. In biological organization, you have different level of organization of proteins. As I told you, sir, empty slide. As I told you in the biological organization, there are different levels of organization of proteins. The first and the foremost is what structure? The primary structure, where you have one amine group and what? Carboxylic acid. This is linear structure and this is primary. The next we have the secondary structure. The next we have what structure? Secondary structure, wherein secondary structure we find, okay, the same linear structure could coil, okay, in a right-handed helix, in a right-handed fashion, and it is called as alpha helix. This is one of the pattern of secondary structure called as alpha helix, or it can arrange in a zigzag manner. It can arrange in a zigzag manner called as what? Beta pleated. These are the two different forms of secondary structure. What forces work here? In primary structure, the forces are peptide bond. In the secondary structure, the forces are, as you have studied in the chemistry part, covalent bond, ionic bond, electrostatic, all weak forces including strong, van der Waals, right? All these are the different forces that will hold the alpha helix and the beta pleated sheets together. This is secondary structure of organization of protein. Okay. Next, we have two more organizations. We call it as tertiary structure. So tertiary structure is one single polypeptide. Tertiary structure is what polypeptide? One single polypeptide where amine group, it will coil and carboxylic acid. It appears like a woolen ball and it will have a biological role as an hormone or an enzyme. It will have a biological role as an hormone or an enzyme. Finally, quaternary structure. In quaternary structure, example is hemoglobin pigment made up of 4 polypeptide chains, alpha 1, alpha 2, beta 1, beta 2. Then only it will be functional if it has all the 4 polypeptide chains. So this is alpha 1, NH2, COH. This is one polypeptide chain, alpha 1, and this is alpha 2, NH2, COOH, alpha 2. Then this is beta 1, COOH, NH2, which is beta 1. Then this is beta 2, NH2 in yellow color chalk, COOH, beta 2. Okay. So more than one. For hemoglobin, how many polypeptide chains are there? 4 polypeptide chains. Okay. To act as an enzyme or a hormone. To act as an enzyme or what? Hormone. So there is a major difference between tertiary and quaternary structure. Here it is one polypeptide chain. Here it is more than one. But tertiary and quaternary have a biological role to act as an enzyme or what? Hormone. These are the 4 different levels of organization of proteins for biological role. Hope it is clear for you. Any doubts, you please ask me. Done? Okay. So finally, to conclude with proteins altogether, we say homopolymer. What is homopolymer? Made up of only one type of amino acid, like lysine, lysine, lysine. If the whole polypeptide chain is made up of one single specific of amino acids, it's called as homopolymer. But such homopolymers are not generally found. Okay, and the next is the heteropolymer. In heteropolymer, lysine, glycine, valine. So they are generally found in animal tissues and plant tissues. And again, finally, essential and non-essential. Essential should be taken through diet. Non-essential can be synthesized in the body. Non-essential proteins can be synthesized in the body itself. Hope this all regarding the structure of proteins. Understood, students? Yes, sir. Slide, please. Sir, slide, please. So what are proteins? They are the polymers of amino acids. Homopolymer containing one type of monomer. Heteropolymer. Next. Next. Having different types of monomers joined through peptide bond. Next. What are essential? Should be taken through food or diet. Non-essential, where the body can synthesize. Abundant protein in animal world is collagen. Abundant protein in the whole biosphere is the enzyme rubisco. Is the enzyme rubisco found in what tissues? Plant tissues. Next. See, these are some of the examples of proteins and their function. Collagen, intercellular brown substance, trypsin, enzyme, insulin, hormone, antibody fight infectious agents, receptors for sensory, smell, taste, and hormone, GLUT4, enable glucose to mobilize into the cells. Again, it is very important for NEET. Next. Next. So in the primary structure, it is a linear arrangement of amino acid from first to last. In the first amino acid, what is exposed? NH2. In the last amino acid, what is exposed? Carboxylic acid. That is the reason we write NH2COOH for primary structure. Yes, sir. Next. Secondary structure to form alpha helical or beta pleated. Next. See here. This is primary, linear, alpha helical, beta pleated. Tertiary, generally made up of one polypeptide chain. Quaternary, made up of more than one polypeptide chain. Quaternary, made up of what? More than one polypeptide chain. Next. So tertiary structure, the long protein chain folded upon itself to have a woolen ball-like structure. Quaternary, more than one polypeptide chain with individual foldings with respect to each other called as quaternary structure. Next. See, this is one single polypeptide with disulfide bonds that holds together. These two are secondary structure and this is tertiary structure, one single polypeptide chain. Next. See, this is quaternary structure for hemoglobin pigment. See, 4 chains, alpha, alpha, beta, beta. Next. So carbohydrates. Sir, I think time is up. Sir?
Yes, sir. Yes, sir. So it is 12:30 now, sir.
Yes.
So do we do this in the next class, sir?
Yes, sir. Carbohydrates and regarding enzymes and its functioning.
Yes, sir.
We can do it in the next class.
Okay, sir. So next class scheduled is almost same time, students, on Thursday.
Yes, sir. Sir, before we conclude, you want to ask me some questions?
Please, please, sir.
Okay, sir. You can ask anyone student on.
Yes. Yes, Levin. Unmute yourself, no?
Yes, sir.
Yes, sir. You can
Okay.
ask.
Okay. Next. So, what is a nucleotide?
Sir,
Nucleotide.
so nucleotide is the nitrogenous base, sugar, and phosphate.
Okay. What a backbone of DNA is made up of?
So, sugar and phosphate.
Good. What is a base pair?
Sir,
Base pair.
I am not sure, sir.
I told you Chargaff rule, right?
Oh, the adenine double bond hydrogen with thymine.
Yes,
That one?
that is a base pair. That is one base pair. Which is other base pair?
Guanine with triple hydrogen bond with cytosine.
Okay, very good. Next. Sir, any other student?
Evelyn.
Yes, sir.
Okay, ma. Can you name some secondary metabolites?
Secondary. Sir, I'm not sure, sir.
So I told many examples. Can you name some? We also studied the table of secondary metabolites also.
Essential oils,
Yeah. Any other clue?
alkaloids,
Hmm.
alkaloids, and
Terpenoids,
yes,
right? Okay. So what is the function of secondary metabolites?
defense mechanism for plants.
Okay, good. What is a homopolymer?
Made up of one type of monomer.
Yes. Heteropolymer?
Made up of more than one monomer.
Okay, which is the most abundant protein in the whole of biosphere?
Rubisco.
Okay, very good. Done. Yes, sir.
Yes, sir, Anika.
Yes, sir.
Okay, ma. What are essential amino acids?
As in the one which are obtained from diets and fruits.
Yes, you have to obtain it from dates and fruits. So, what is the percentage of proteins in a total cellular mass? We studied in the table.
Sir?
We studied in a table, and I told the table is very important. What is the percentage of proteins in a cell?
Is
Okay,
it
difficult to read.
20%, sir?
10% to 15%. We have table in the NCERT textbook. Please go through it. Okay.
Yes, sir.
So again, to repeat the same, in which fraction do we find the lipids? Acid soluble or acid insoluble?
Is it acid insoluble, sir?
Yes, acid insoluble fraction, because they form micelles and they get trapped within the pellet, so that it precipitate in what fraction? Acid insoluble fraction. Yes or no?
Yes, sir.
Okay, ma. Can you name any one nucleotide?
Thymalic acid, sir.
Okay, any other?
Adenylate acid.
Okay, great. Fine. Good. Okay, sir.
Yes, sir. Any more questions, sir?
Yeah, yeah. Done.
Done, sir.